Welcome to Proteomics Unlocked, where we explore how protein-level analysis advances precision medicine and biological research. This opening slide sets the stage with images of protein structures, data visualizations, and analytical workflows, reminding us that proteomics connects molecular detail to actionable biology and clinical insight across discovery and translational pipelines. Proteomics goes beyond genomics by bridging transcriptomics and physiology, showing that mRNA levels often poorly predict protein abundance. This slide highlights the mRNA-protein disconnect driven by translation control, degradation, and post-translational modifications, and emphasizes proteins as the primary cellular actors that catalyze reactions, shape structure, and transmit signals, which demands direct protein measurement. Here, we overview core proteomics technologies, from ionization methods like ESI and MALDI, to mass analyzers such as quadrupole, Orbitrap, and time of flight, and the key metrics of resolution, mass accuracy, and dynamic range. The diagram also shows tandem MS sequencing, where precursor fragmentation generates peptide fragments used for identification and characterization. Peptide separation and complexity reduction are essential to detect low-abundance species, and this slide covers nano-LC reversed phase gradients, orthogonal dimensions like capillary electrophoresis and ion mobility, and sample pre-fractionation strategies. It also discusses the trade-off between peak capacity and run length, and how reducing matrix complexity boosts sensitivity for rare peptides. Turning raw spectra into reliable abundance metrics requires careful processing, MS1 and MS2 proxies, retention time alignment, and normalization across runs. The slide contrasts DDA and DIA acquisition modes and underlines the importance of quality control and batch effect mitigation so that comparative studies yield reproducible, biologically meaningful results. Stable isotope labeling and multiplex techniques enable comparative proteomics at scale. Methods like SILEC, isobaric tagging with iTrack, or TMT, and cost-effective dimethyl labeling each offer ways to multiplex samples, compress ratios, and improve throughput. The slide highlights how these approaches streamline workflows, reduce variation, and support clinical and time series studies. Phosphoproteomics maps signaling by enriching phosphopeptides with TiO2 or IMAC, precisely localizing serine, threonine, and tyrosine sites via MS, and quantifying stimulus-induced phosphorylation over time. The workflow leads into network reconstruction, integrating kinase-substrate relationships to model cellular responses to drugs, growth factors, or stress conditions, and reveal pathway-level biology. Targeted modification workflows focus on glycosylation and ubiquitination, describing N and O-linked glycans that affect folding and recognition, and ubiquitin remnant profiling to probe protein degradation. The slide points out specialized enrichment strategies, such as lectin affinity and enzymatic release, and highlights the biological impacts on immune response, oncogenesis, and protein half-life. The diagnostic workflow captures the path from case control profiling through statistical feature selection to targeted validation and clinical translation. It outlines large-scale sample acquisition, biomarker discovery with ML, confirmation by SRM or PRM, and assay transfer to ELISA or CLIA platforms, stressing independent cohorts, blinding and clinical utility assessment for robust adoption. Quantifying drug target engagement combines thermal shift assays, chemical proteomics, and affinity pull-down to measure binding, occupancy, and off-target effects in cells. This slide describes dose-response curves, off-target identification by mass spectrometry, and how these approaches feed lead optimization and support selection of clinical candidates with improved specificity and safety. Single-cell proteomics unlocks cellular heterogeneity by using nanoscale sample prep carrier channels and highly sensitive Orbitrap instruments to quantify thousands of proteins per cell. Noise reduction and enhanced quantification accuracy are essential, and integrating these data with single-cell transcriptomics helps map functional networks, tumor or immune heterogeneity, and direct functional readouts. AI-driven acceleration in proteomics brings deep learning predictors, automated de novo sequencing, and real-time adaptive acquisition together with cloud-based spectral libraries. The slide shows how these technologies speed protein identification, improve quantification accuracy, enable dynamic sampling and collaborative sharing, and ultimately drive faster hypothesis generation and large-scale discovery.